Hi, my name is Yang from Northwestern University in Chicago, and today I'm going to talk about multi-slice 2D flow MRI for cerebrospinal fluid flow as a strategy for improved SNR and velocity noise. CSF is clear and colorless body fluid found in the brain and spinal cord. It is produced in the coronal plexus of the ventricles of the brain, and it usually produces 500 milliliter per day. The CSF circulates the brain and spinal cord, and then it is absorbed in the erythroid granulations into the vessels. The main function of CSF for our body is to act as a cushion or buffer, providing mechanical and immunological protection to the brain. So, how the CSF flow is related to neurological disease? Several studies have shown that changes in CSF flow dynamics have been found to be associated with different neurological disease. One example of hydrocephalus child, since the adductor passage between lateral and third ventricle is blocked, the CSF in the ventricles gets bigger, so CSF flow in the hydrocephalus have found up to a tenfold increased pulsatile aqueductal flow compared with the health controls. Other studies have shown that the Chiari malformation can restrict normal CSF flow between the cranial and spinal compartment that in turn could result in sirens. So the quantification of the CSF flow is necessary for a better understanding of the physiology and pathophysiology of CSF dynamics. It may also assist in diagnosis and treatment guidance of the CSF-related disease. For this purpose, a body flow MRI has been a useful technique to visualize the flow field and to investigate the effect of abnormal CSF flow on the neurological disease. One recent studies have revealed that the velocity mapping with MRI machine is applicable to studying flow patterns of CSF in the human ventricular system and allows for clarification of the patterns of the CSF flow. Other studies showed that the 4D flow MRI allowed comprehensive analysis of CSF flow in patients with Chiari I malformation, visualizing pronounced alterations of CSF hydrodynamics. 4D flow MRI has been a promising technique for CSF flow dynamics, but due to its low signal intensity, the acquisition of 4D flow MRI dataset is accompanied with low SNR and high velocity noise. The main reason of low signal when scanning with conventional 4D flow MRI is that the CSF has a significant long T1 relaxation time than its short repetition time of 4D flow MRI scans. So, how do we increase the signal intensity? One simple idea is that we can give more repetition times to each slice by adjusting the RF excitations order as shown below. For example, if we set the slice encoding to be interleaved in the 2D flow MRI sequence, we can give more repetition times to each slice. So my research objective is to investigate a strategy of multi-slice 2D flow MRI acquisitions for CSF flow measurement in terms of signal-to-noise ratio and velocity noise. For this purpose, the sequence is designed to give longer repetition time for each slice acquisitions. In vitro test was performed to investigate the performance of the sequence and compare with the conventional 2D and 4D flow MRI sequence. Here is the comparisons of the conventional 4D flow MRI sequence and the new sequence design for CSF flow measurement. As you see the figure above, the conventional sequence can give short TR at slice 1 acquisitions for each velocity encoding. But the new sequence can get more repetition time than conventional sequence for each slice acquisitions at each velocity encoding. For example, when you set to get 7 slices acquisitions, we can get 7 times longer repetition times than a conventional sequence. This strategy could be useful for multi-slice 2D flow MRI acquisitions. The MR parameters for in vitro tests are summarized in this table. For comparisons, the conventional 2D and 4D flow MRI sequences are used. For 10 slices scanning, the repetition time of CSF sequence is set to be 100 milliseconds, while conventional sequence has a repetition time of 8 milliseconds. Also, several scans are performed by varying flip angle to investigate the optimal flip angle with the highest signal intensity. For the in vitro test, 1.5 Tesla era MRI machines with body core is used. I also used water which has T1 of approximately 3000 milliseconds. Although there are slight differences of T1 between CSF and water, but I think T1 of water is long enough for this test. As analysis phantom, tubing filled with water was used and 2 barrels of dummy water phantom were inserted for signal acquisitions. And I'm going to show you the result. These are the representative magnitude image measured by the conventional 2D and 4D sequence and CSF sequence. As you see, the conventional sequence gives bright image at flip angle of 10 degrees and the CSF sequence gives at flip angle of 20 degrees. This is because the optimal angle changes as the repetition time of the CSF sequence is changed. We can also check that the CSF image at optimal angle of 20 degrees is brighter than the conventional image at optimal angle of 10 degrees. For quantitative analysis, I've plotted the mean value and standard deviation of 10 slices data. The SNR is estimated as a signal magnitude by background noise. The velocity noise is also estimated as a standard deviation of velocity measurement data. The result shows that the optimal angle for each sequences were estimated as 10 to 12 degrees for a conventional sequence and 20 to 25 degrees for CSF sequence, when looking at the maximum signal-to-noise ratio and minimum velocity noise. Sequence comparisons at each optimum flip angle shows that the SNR of CSF sequence gives 140% increase than conventional 2D sequence, and velocity noise of the CSF sequence also gives 33 to 47% decrease than conventional sequence. With the result of this new sequence for CSF flow measurement, we are expecting to evaluate the feasibility of in vivo CSF flow measurement and investigate the effect of abnormal CSF flow in disease as a future study. And this is the conclusions. The strategy of multi-slice 2D flow MRI gives improved SNR and velocity noise than the conventional 2D and 4D flow MRI. Future study will include evaluating the feasibility of CSF flow measurement and investigating the effect of the abnormal CSF flow in disease. And this strategy to give longer repetition time acquisitions is limited to 2D multi-slice flow MRI because of the necessary rearrangement of slice encoding ordering. And thank you for listening.